So in this video, we are going to discuss about back cross. So in the previous video, we have already discussed about test cross. So in genetics, there are mainly three crosses are there. Test cross, back cross and reciprocal cross. So test cross, as we have discussed that it is very important to know the genotype of F1 hybrid. So today we are going to study back cross. The name back itself indicates that it is a cross which is done with something with back. So here it is defined as a cross between F1 hybrid with either of the two homozygous parents. Let us see by taking an example. So now as we know that we have studied with an example monohybrid cross. So which is a cross which is obtained between that is tall plant and dwarf plant. So here also we are going to discuss about same thing now. So now imagine now when a tall plant homozygous, so just the parents, we are going to write here the parents. So now coming to the parents, we can find that a cross is made between homozygous tall and homozygous dwarf. So homozygous tall, okay, with homozygous dwarf, as we have already learnt many times, homozygous dwarf. So when this cross is done, so genotype of this, so when we see the genotype of this homozygous tall, we can write it as capital T, and the dwarf is represented by small t because it is recessive in nature. And then we are going to get a gametes. So here small capital T and here the gamete is small t. Gamete means we know which is haploid in nature. So next, when it undergoes fertilization in F1 generation, we are finding that when, imagine now this is a pollen grain, when the pollen grain fuses with the egg, the genes obtained here is capital T and small t. So this is our F1 generation. We are getting heterozygous tall. We got here as heterozygous tall we have got. So now we are going to do back cross for this. So please understand this. Now we are doing back cross. So as the definition itself indicates that it is a cross between, you can find here F1 hybrid is crossed with either of the two parent. Okay. Now which are the parents? So here tall is a parent, dwarf is a parent. So capital T and small t are the homozygous parents they are. Now this is F1 hybrid. So back cross is nothing but F1 hybrid is crossed with, okay, any of the parent. Now we are taking any parent we have to take. Either it is dominant parent or recessive parent. So now F1 hybrid is, this is our F1 hybrid. So I am taking here capital T, small t. Then any parent. So I am taking here this capital T, that is dominant character I am taking here. So dominant character I am taking capital T. So now here we know that the gametes obtained here is, so here we are going to get capital T and small t. These two are the gametes. Same like this you have to remember. Now genotypes we have written. These two are the gametes. In this case we can find that, so the gametes here includes capital T and capital T. So now if we do a checkerboard for this, if you do the checkerboard for this, we know that we are going to get the one side we are writing here it as, you know very well. So male and female, here capital T, small t and T, capital T. So if a cross is made between capital T and capital T, here you are getting TT. and this capital T with small t, capital T, small t we get. Here also you are going to get capital T, and when this T is crossed with this, then you are going to get capital T, small t. So here we are going to get 2 homozygous tall and 2 heterozygous tall. So all plants obtained here is, all plants obtained are tall. You know very well, all plants obtained are tall. So this is nothing but homozygous tall. These two are homozygous tall, whereas these two are heterozygous tall. Heterozygous tall. So whatever may be, you have got tall plant. Even back cross, now you came to know. So this F1 hybrid is here. This is either crossed back. The name itself is telling either you are crossing this with back to the homozygous tall, or you can cross this F1 hybrid back to the homozygous dwarf. So let me see, in case of homozygous dwarf, what genotype we are going to obtain. So now in the same case, instead of same thing, this capital T, small t into, now I am taking this as a parent. So small t, small t. If I take like this, here also I am getting the gametes, capital T and small t I am getting gametes, and in this case I am getting small t and small t. So if I do a checkerboard for this cross, even I am going to get all, we just see what exactly the genotypes we are going to get. 1 second. Okay, I will erase this. Okay, so in this case, let us see what exactly the genotypes we are going to get. So if I take checkerboard for this, so for this cross, see the difference, what difference we are going to get here. So capital T, small t, and here also small t, small t. So in this case you are getting capital T, small t, here you are getting small t, small t. This is nothing but capital T and small t, here you are getting small t, small t. So in this case you can see you have got heterozygous, 2 heterozygous tall you got, heterozygous tall plants you got, and 2 dwarf plants you got, homozygous dwarf plant you have got. So in this cross if you do, you are getting this. In this cross if you do, you have got all tall plants. Okay. So now we are going to understand what is the significance of this back cross. So as we have seen in the back cross, the significance when you see, we can tell that it increases, it is done in order to increase the number of traits of the parent. See now, if you want all tall plants, then you have to do back cross with dominant tall plant. If you want dwarf plant, you can do a cross with a recessive parent. If you do cross with recessive parent, you are going to get tall and dwarf. But if you do it with a dominant parent, you will get all tall plants. If you go on repeating the back cross, you will get pure tall plants, homozygous tall plants, or you may get pure heterozygous tall plants. Usually to get homozygous tall plants, you are going to do back crosses. Okay. So next thing is, it is very useful in case of horticulture and animal breeding, and it is used in order to knock out certain genes. Now in knockout means what? See, in that if I do back cross, I may get heterozygous tall. But in order to avoid this heterozygous tall, if I do back cross, I am going to get always pure homozygous tall I can get if I repeat the experiment. Okay. So next we are going to study about, we can find here, that is the difference between test cross and back cross. So let us see what is the difference between test cross and back cross. Very, very important it is, and it is a slight difference is there. You have to understand it properly. So it is a cross between F1 hybrid and its recessive parent. So last previous video only we have explained that F1 hybrid is crossed with recessive parent. Okay, recessive parent. Then it is called as test cross. And understand this, when it is crossed with recessive parent, it is called as test cross. But when a F1 hybrid is crossed with any one of the parent, either it is dominant or recessive, then it is called as back cross. Second thing is, a test cross is always a back cross. So we have already understood. So test cross always is crossed with recessive parent. So back cross. So here the back cross is not always a test cross. Why? Why? Because back cross is also done with the dominant parents. If it is done with recessive parent, then it is called test cross. But if F1 hybrid is done with a dominant parent, it is not called as a test cross. Then next we can say test cross produce. So I have done in the last video, that is when we do test cross, we are going to get both dominant and recessive characters. We used to get 1:1 ratio. Whereas in case of back cross, if you do back cross with a dominant parent, just now we have seen all plants which are obtained were dominant only. But if you do with a recessive parent, you are going to get heterozygous tall and even recessive character also you can see. The next one is, it is used to understand the law of segregation. So if you do completely this test cross, you have seen that capital T and small t are getting segregated. And in this back cross, we can see how the genes has been passed from one generation to another generation. That is nothing but to verify the law of inheritance. Next, we know that test cross determines the genotype. So this I have already told you, very, very important. If you do the test cross, you will be knowing whether the F1 hybrid is homozygous or heterozygous. So this back cross, it helps in getting what we can say required characteristic features. If you want tall plants, always you can go for dominant parent itself. Okay, that is what we can say, that the significance of back cross it is. So in the next we are going to study what is reciprocal cross. So reciprocal, the name itself indicates that reciprocal means reverse. Okay, so now it is a cross which is involved, two crosses are involved in this. So it involves two crosses. Let us see what are the two crosses. So it is nothing but a cross which is involved in reversing the sex of the organism. Please remember this. So with the reversed sex. So if you are considering two plants, imagine now, for example, I am telling you. So here we always crossed with, tall plant, and tall plant is crossed with dwarf plant, right? So imagine now this tall plant is considered as a male plant. So from this plant you are collecting pollen grains. So small t, imagine it is considered as a female plant. So same thing, in the male the genes of the pollen grains were capital T, capital T. In case of female, the genes in the egg is small t, small t. In this, of course, you know that the parent genes are capital T, capital T. In the pollen grain, you have only one, because pollen grain is haploid in nature. So here that is the female one, it is having the genes diploid in nature, but the egg is having only one, what we can say, haploid small t it is having. When it fuses, you are getting capital T, small t. Here what you are concentrating is, here dominant character you have taken from the male plant. In the next cross, whenever you are doing, you are taking capital T into small t, but now this you are considering it has a female. So you are taking female dominant plant, and male you are taking a recessive plant, you are taking dwarf plant. Dwarf plant is of male and tall plant is from female. But here what happened? It is reversed. So here also same things. The process, everything is same. But what we have reversed here is nothing but the sec of the plant has been, whatever you are collecting has been reversed. Okay. So in the same way, same thing they have given in the example. So if you imagine A is nothing but the, another example it is, if you imagine A is a female and B is a male. So in another example it is, imagine A is nothing but it is a female and B is nothing but the male. Same thing, in the female you are having AA genes, in the male you are having BB genes. So in the gamete you are going to get A and here you are going to get B. When it is fused you are going to get AB. So in the next this one what you are doing is, you are taking A which you are taking it as a male and B you are taking it as a female. That's all. So when you are crossing like this, what happens? You are going to get the result, of course it will be same, but the thing is the sec of the organism has been changed. So what is the importance of this reciprocal cross? Means mainly it is used in order to, mainly we can find that it is used in order to know the role of the sec of a particular characteristic feature, we can say. So now here we can find it that in order to know the role of parental sec of a given inheritance, how particular female character and the male character, how it is going to inheritance and its effect, we are going to understand from this. So in the next class we are going to discuss about dihybrid cross.